Hello! Good day, everyone. This will be our last session for this quarter. Today, we will discuss about the chromosome theory of inheritance and the basic principles on how traits are transmitted. The nuclei of normal human cells carry 23 pairs of chromosomes for a total of 46. There are noticeable differences in size and shape among the 23 pairs, but within each pair, the two chromosomes appear to match exactly. The only exceptions are the male's sex chromosomes, designated X and Y, which constitute an unmatched pair. Down syndrome was the first human genetic disorder attributable not to a gene mutation, but to an abnormal number of chromosomes. Children born with Down syndrome have 47 chromosomes in each cell nucleus because they carry 3 instead of the normal pair of a very small chromosome referred to as number 21. The aberrant genotype, known as trisomy 21, gives rise to an abnormal phenotype, including a wide skull that is flatter than normal at the back, an unusually large tongue, learning disabilities caused by the abnormal development of the hippocampus and other parts of the brain, and a propensity to respiratory infections as well as heart disorders, rapid aging, and leukemia. The chromosome theory of inheritance. It states that chromosomes are the carriers of genes. How can one extra copy of the chromosome that is itself of normal size and shape cause such wide-ranging phenotypic effects? The answer has two parts. First and foremost, chromosomes are the cellular structures responsible for transmitting genetic information. In this lesson, we describe how geneticists concluded that chromosomes are the carriers of genes, an idea that became known as the chromosome theory of inheritance. The second part of the answer is that proper development depends not just on what type of genetic material is present, but also on how much of it there is. Thus, the mechanisms governing gene transmission during cell division must vigilantly maintain each cell's chromosome number. One of the first questions asked at the birth of an infant is it a boy or a girl? Acknowledges that male and female are mutually exclusive characteristics, like the yellow versus green of Mendel's peas. What's more, among humans and most other sexually reproducing species, a roughly 1:1 ratio exists between the two genders. Both males and females produce cells specialized for reproduction, the sperm or eggs, that serve as a physical link to the next generation. In bridging the gap between generations, these gametes must each contribute half of the genetic material for making a normal, healthy son or daughter. Whatever part of the gamete carries this material, its structure and function must be able to account for the either or aspect of sex determination, as well as the generally observed 1:1 ratio of males to females. These two features of sex determination were among the earliest clues to the cellular basis of heredity. In embryonic cells, the chromosomal threads split lengthwise in two just before cell division, and each of the two newly forming daughter cells receives one half of every split thread. The kind of nuclear division followed by cell division that results in two daughter cells containing the same number and type of chromosomes as the original parent cell is called mitosis, which came from the Greek mitos, meaning thread, and osis, meaning formation or increase. In the cells that give rise to male and female gametes, the chromosomes composing each pair become segregated, so that the resulting gametes receive only one chromosome from each chromosome pair, the kind of nuclear division that generates egg or sperm cells containing half the number of chromosomes found in other cells within the same organism is called meiosis, from the Greek word for diminution or diminution. When we talk about fertilization, it is the union of haploid gametes to produce diploid zygotes. Most body cells are diploid. They carry a maternal and paternal copy of each chromosome. Meiosis generates haploid gametes with only one copy of each chromosome. In Drosophila, diploid cells have 8 chromosomes, where 2n is equals 8, while gametes have 4 chromosomes. N is equals to 4. Note that the chromosomes in this diagram are pictured before their replication. The X and Y chromosomes determine the sex of the individual. The corresponding maternal and paternal chromosomes appear alike in size and shape, forming pairs, with one exception, the sex chromosomes. Gametes and other cells that carry only a single set of chromosomes are called haploid, from the Greek word for single. Zygotes and other cells carrying two matching sets are diploid, from the Greek word for double. The number of chromosomes in a normal haploid cell is designated by the shorthand and symbol N. The number of chromosomes in a normal diploid cell is then 2N. Fertilization is the union of haploid gametes to produce diploid zygotes. Metaphase chromosomes can be classified by centromere position. Before cell division, each chromosome replicates into two sister chromatids connected at a centromere. In highly condensed metaphase chromosomes, the centromere can appear near the middle, a metacentric chromosome, very near an end, an acrocentric chromosome, or anywhere in between. In a diploid cell, one homologous chromosome in each pair is from the mother and the other from the father. Cells in metaphase can be fixed and stained with one of several dyes that highlighted the chromosomes and accentuate the centromeres. The dyes also produce characteristic banding patterns made up of lighter and darker regions. Chromosomes that match in size, shape, and banding are called homologous chromosomes or homologs. These two are a pair of homologous metacentric chromosomes, and this is a pair of homologous acrocentric chromosomes. Karyotype of a human male is presented in the image. Photos of metaphase human chromosomes are paired and arranged in order of decreasing size. In a normal human male karyotype, there are 22 pairs of autosomes, as well as an X and Y chromosomes. Homologous chromosomes share the same characteristic pattern of dark and light bands. To study the chromosomes of a single organism, geneticists arrange micrographs of the stained chromosomes in homologous pairs of decreasing size to produce a karyotype. Karyotype assembly can now be speeded and automated by computerized image analysis. Pairs are known as autosomes. The two unmatched chromosomes in this male karyotype are called sex cells, or sex chromosomes, because they determine the sex of the individual. Walter Sutton, a young American graduate student at Columbia University in the first decade of the 20th century, was one of the earliest cytologists to realize that particular chromosomes carry the information for determining sex. In one study, he obtained cells from the taste of the greater lubber grasshopper or Brachystola magna and followed them through the meiotic divisions that produce sperm. He observed that prior to meiosis, precursor cells within the testes of a great lubber grasshopper contain a total of 24 chromosomes. Of these, 22 are found in 11 matched pairs and are thus autosomes. The remaining 2 chromosomes are unmatched. He called the larger of these the X chromosome and the smaller the Y chromosome. This figure shows the great lubber grasshopper. In this mating pair, the smaller male is astride the female grasshopper. Species variations in sex determination. You have just seen that humans and other mammals have a pair of sex chromosomes that are identical in the XX female but different in the XY male. Several studies have shown that in humans it is the presence or absence of the Y that actually makes the difference. That is, any person carrying a Y chromosome will look like a male. For example, rare humans with 2 X and 1 Y chromosomes are males displaying certain abnormalities collectively called Klinefelter syndrome. Klinefelter males are typically tall, thin, and sterile, and they sometimes show mental retardation. That these individuals are males shows that 2 X chromosomes are insufficient for male development in the presence of a Y. In contrast, humans carrying an X and no second sex chromosome are females with Turner syndrome. Turner females are usually sterile, lack secondary sexual characteristics such as pubic hair, are of short stature, and have folds of skin between their necks and shoulders. Even though these individuals have only one chromosome, they develop as females because they have no Y chromosome. This figure shows how the X and Y chromosomes determine sex in humans. Figure 4.6a shows the colorized micrograph of the human X chromosome and the human Y chromosome. In Figure B, children can receive only an X chromosome from their mother, but they can inherit either an X or a Y from their father. The cell cycle, an alternation between interphase and mitosis. Chromosomes replicate to form sister chromatids during synthesis or S phase. The sister chromatids segregate to daughter cells during mitosis or the M phase. The gaps between the S and M phases, during which most cell growth takes place, are called the G1 or GOP1 and G2 phases. In multicellular organisms, some terminally differentiated cells stop dividing and arrest in the G0 stage. Interphase consists of G1, S, and G2 phases together. This figure shows the mitosis maintains the chromosome number of the parent cell nucleus in the 2 daughter nuclei. In the photomicrographs of newt lung cells at the left, chromosomes are stained blue and microtubules appear either green or yellow. So we will have a discussion on the different phases of mitosis. Let's start with the first stage, which is prophase. So the chromosomes condense and become visible. The centrosomes move apart toward opposite poles and generate new microtubules, and the nuclei begin to disappear. In the prometaphase, the nuclear envelope breaks down. The microtubules from the centrosome invade the nucleus, and the sister chromatids attach to microtubules from opposite centrosome. In the metaphase stage, the chromosomes align on the metaphase plate, or sometimes called the equatorial plane, with sister chromatids facing opposite poles. In anaphase, the centromeres divide. The now separated sister chromatids move to the opposite poles. The telophase, the nuclear membranes or nuclei reform, and the spindle fibers disappear. The chromosomes uncoil and become a tangle of chromatin. And the last stage, which is the cytokinesis, the cytoplasm divides, splitting the elongated parent cell into two daughter cells with identical nuclei. These two images shows the cytokinesis of the parent cell producing two daughter cells. So this figure shows the summary of meiosis. The chromosomes replicate once while the nuclei divide twice. All four chromatids of the chromosome pair are shown in the same shade of the same color. Take note that the chromosomes duplicate before meiosis 1 and do not duplicate between meiosis 1 and meiosis 2. This image shows the difference between mitosis and meiosis. So mitosis produces two daughter cells identical to each other and in the original cell. Mitosis is thus genetically conservative, whereas meiosis produces four haploid cells, one egg or all sperm, of which can become gametes. None of this is identical to each other or to the original cell, because meiosis results in combinatorial change. Gamete formation or gametogenesis thus gives rise to haploid gametes, marked not only by the events of meiosis per se, but also by cellular events that precede and follow meiosis. Here, we illustrate gametogenesis with a description of egg and sperm formation in humans. Oogenesis in humans produce one ovum from each primary oocyte. The end product of egg formation in humans is a large, nutrient-rich ovum whose stored resources can sustain the early embryo, the process known as oogenesis, as shown in this figure. This begins when diploid germ cells in the ovary called oogonia multiply rapidly by mitosis and produce a large number of primary oocyte. Okay, which then undergo meiosis. For each primary oocyte, meiosis 1 results in the formation of two daughter cells that differ in size. So this division is asymmetric. The larger of these cells, the secondary oocyte, receives over 95% of the cytoplasm. The other small sister cell is known as the first polar body. During meiosis II, the secondary oocyte undergoes another asymmetrical division to produce a large haploid ovum and a small haploid second polar body. The first polar body usually arrests in its development and does not undergo the second meiotic division. However, in a small proportion of cases, the first polar body does divide, producing two haploid polar bodies. The two, or rarely three, small polar bodies apparently serve no function and disintegrate, leaving one large haploid ovum as the functional gamete. Thus, only one of the three products of a single meiosis serves as a female gamete. A normal human ovum carries 22 autosomes and an X sex chromosome. From the onset of puberty at about age 12 until menopause, some 35 to 40 years later, most women release one primary oocyte each month from alternate ovaries. Take note, alternate ovaries, amounting to roughly 480 oocytes released during the reproductive years. The remaining primary oocytes disintegrate during the menopause. Spermatogenesis in humans produce 4 sperm from each primary spermatocyte. The production of sperm or spermatogenesis begins in the male testis in the germ cells known as spermatogonia. Mitotic divisions of the spermatogonia produce many diploid cells, the primary spermatocytes. Unlike primary oocytes, primary spermatocytes undergo symmetrical meiosis I, producing 2 secondary spermatocytes, each of which undergoes a symmetrical meiosis II. At the conclusion of meiosis, each original primary spermatocyte thus yields 4 equivalent haploid spermatids. These spermatids then mature by developing a characteristic whip-like tail and by concentrating all their chromosomal material in a head, thereby becoming functional sperm. A human sperm, much smaller than the ovum it will fertilize, contains 22 autosomes and either an X or a Y sex chromosome. The timing of sperm production differs radically from that of egg formation. The meiotic divisions allowing conversion of primary spermatocytes to spermatids begin only at puberty. But meiosis then continues throughout a man's life. The entire process of spermatogenesis takes about 48 to 60 days, 16 to 20 for meiosis I, 16 to 20 for meiosis II, and 16 to 20 for the maturation of spermatids into fully functional sperm. Within each testis after puberty, millions of sperm are always in production, and a single ejaculate can contain up to 300 million. Over a lifetime, a man can produce billions of sperm, almost equally divided between those bearing an X and those bearing a Y chromosome. I guess that's all for this session. A detailed process of meiosis and mitosis will be discussed in the next session. See you in our midterm examination. Thank you. God bless. Bye.